Hello everybody, let's do the video for 3.5 now. So these blanks, I'm going to leave them in there so I don't lose my place. Remember, we're going to use, I want you to figure out these blanks as best you can on your own. I'll give you stamps the more blanks you fill in, and I'll give you a key before the exam. But let's get into gel electrophoresis, because you all have been doing electrophoresis just right now. You see a familiar setup here. We have a power supply right here. As we know, 300 volts is not cool. You want to keep it about 115 volts. Definitely. We had melted that gel this year. Kind of cool though. Kind of cool. Anyway. Black, as we know, is for negative. Red over here is positive. And if we're dealing with a negatively charged molecule like DNA, it's going to move towards the positive. Not everything is positive. So when I say direction of flow or movement, I'm talking about DNA and any negatively charged particle. That could be a protein, dye, whatever. Of course, as you remember, we need to put a solution in there. That's called a buffer. And that helps channel the electricity from one side to the other. And these holes in the gel are called wells. Those are the holes we're going to put our DNA product in. And remember, there's three things you can tell by using gel electrophoresis. One is the relative size. Should I fit it in here? Relative size of fragments. Larger equals shorter travel distance. Smaller size of particles equals travels further. All right. The second thing is you can figure out the charge of the particle, right? Because we know positive ones go towards the negative, negative go towards the positive. So we can tell the charge of particle.
That's a wrap.
And in relationship to the dye lab that we do, the gel electrophoresis dye lab, we figure out the number of pigments in the dye. It's the number of pigments that the dye is made out of. For example, the green one should be yellow and blue. Okay, so the number of pigments. Another critical technique used in molecular biology is PCR. What we were doing that today. Trying to wrap it up at the end of the year. But in PCR, you have some components. You have the DNA that we're getting from you. Then you have primers. In our case, RNA, or in most cases, RNA primers. And then you have nucleotides. So this Taq polymerase, instead of DNA polymerase, it's this Taq polymerase. And if we put regular your body DNA polymerase in there, it would just disintegrate because it's not used to the high temperatures. This is a special polymerase that's been developed using bacteria that live in really hot thermal vents or pools, and it can withstand the heat, so to speak. So you put all those mixture and you put it in the thermal cycler. Remember the one that we have in class, $5,000. Woo! It's like a car. Ha! Don't be selling it on OfferUp. And I was taking you through. I had you come over, show the process, or explain. We have a heat up almost to boiling. That is the denaturation. remember, in the human body, that's performed by the helicase, right? But we don't want to worry about helicase here. We just want to separate it. We have the heat do the job. Then cool it down quickly to 55, which is like 130 degrees, pretty hot. And this is where you have the annealing of the primers. It's called annealing, so the primers attach. Like it says right there, primers attach. And then you have heating up to 72, like 160, 170, I think. Synthesizing the new strand, that's called extension. It wasn't that the rapper that died a while back, Extensacion, I can't. Anyway, it was a big deal when he was tragically killed, but I just thought of that just now. But remember those 3 steps are repeated 30 times, so you get millions of copies. I forgot the exact number. Might be billions. I gotta check that. Sorry, I don't have that number on me. So that's PCR in a nutshell. You guys have been doing it. I'm not aware of any other high school class in the district that does this, that does PCR or the sequencing that we're going to do. Okay, so ethical issues, you go through that. Now we're kind of on to application and skills, and you all have heard about DNA profiling, identifying people based on their DNA letters, paternity testing, all those sorts of things that are used all rely on gel electrophoresis and PCR. So when you learn about this right now, and you're learning about stuff that half of the TV shows are made out of. Not when I was your age, there were Maury Povich, Jerry Springer, Jenny Jones. Those 3 did almost totally paternity stuff. Almost totally who's the dad and stuff. Anyway, it was not as bad anymore, but there's other sorts of things that are bad. Anyway, CODIS, these are 13 loci, locations in the human chromosome. You know, they're used for identification, used for ID. And all these things, they don't have anything to do with how your body, so they're not junk DNA, but they are not used, they're not used to make any proteins that we know of. Like, these aren't genes. These are just locations that scientists have discovered are highly variable between people. Like, people tend not to have these be the same because just randomly they're different. Highly variable. So they're good to use to identify people. Like, for example, you do not want to use the insulin gene to figure out who murdered somebody if they left DNA behind, because everybody pretty much has the same letters. So everybody's going to match that. The gene for brown eyes, everybody going to match that. The gene for a liver enzyme, pretty much everybody's going to have the same gene. You got to choose things that make us different. And by choosing things that make us different, you can then go to identification. So here is a gel electrophoresis. We have some bands here and sexual assault case. And let's see, we have the victim right here. And this is showing the victim's bands. We have different suspects, 1 and 2, and then the boyfriend. Okay, now it's the victim. What we have here, this is what we need to match it with, the sperm DNA found at the crime scene. So boom, we want to look at those alleles that are found. We just want to look across. If we look here, we see a dead match for suspect 1. So suspect 1 did it, the boyfriend did not. So did the boyfriend do it? No, it was suspect 1. Not that difficult. You're just matching. Well, I hate saying that. I shouldn't say that. You just have to match up. It does look kind of difficult initially. I gotta take that back. It's terrible to say that kind of thing. What you're looking for are the matches. Those bars, those gels, those lines in the gel to match up. Let's practice this one. So here we have a mother, and by the way, ladder is just there to give you known lengths. So this is a ladder where all these marks that you're seeing, all these bands are of a known length for the number of A's, T's, C's, G's, like 300, 3,000, 800, however it is. So it kind of gives you, the researcher, a way to measure. So here's the mother. DNA, here's the child, right? Matches with the mother. So whoever's going to match this is going to be the father. Okay? So if we look here, alleged father, no matching. Now, whoever this is, see a child, a alleged father mix. So mixing them together, I guess. I'm not sure why they would do that, but we don't see that there. So it's almost pretty close. Like this is pretty close to here, but it's not quite matching. So the father doesn't have any, like there's nothing, here's nothing. It's not matching up. Now down here, here's the child. Got these two. And here's the alleged father. Ooh, here. Yeah, right there we know. Here, father is yes. Top one is no. And this is something that's used in paternity tests, this probability of paternity. You may have seen that in court cases. There's a 99.99982% chance that the suspect or the defendant is, DNA matches the DNA found at the crime scene. So you're just looking at the alleles that show up. The alleles that the father has, do they show up in the child? And yes, they do. Yes, alleles seem to match. And you can see that here in the probability of paternity. It's really high. And this is done in some cases in like missing kids. Fortunately, like kids found, they can't be like in explosions or something, terrorist things, wars, when all you have is DNA evidence. School shootings, unfortunately, my God, they might do a profile like this to try and match it with potential parents, to match the child with potential parents. And this next couple slides talk about really cool technology, transferring genes from, say, one plant to another plant, or transferring genes from an animal to a plant. All that stuff can be done, and it's really mind-blowing. And it all comes down to these things called restriction enzymes. And what they do, they know exactly where to go and make certain cuts in the DNA. Like here, you have this cut, and when you cut the DNA, you get these things called sticky ends, and then you can insert other pieces of DNA right by it. So the new DNA, it'd be like a gene, would be placed here. And then the organism could then go ahead and read that DNA and make whatever protein that it codes for. Pretty wild. This is what people talk about with genetic engineering, non-organic foods primarily. They're talking about foods that have used this kind of a technique to change them. Humans have always modified are animals and plants, but that's a slow process through artificial selection. This is a very fast process, and now it's even faster with a technology called CRISPR, which hopefully, well, we're gonna do it next year. We're gonna do it 2022, 2023. There's a kit that I can buy now that takes us through the CRISPR technique, which I think is bad. It's bad. It's awesome. Bad meaning good. Oh, and here it kind of gives here another diagram. This is the DNA to be inserted, and you can really change the phenotypes and genotypes of organisms doing these kind of things, using these kinds of techniques. And there's a whole bunch of different kinds. These are all different kinds of restriction enzymes.
That's a wrap.
Restriction enzyme. And they all cut different places, you know. And by doing that and knowing that, and really there's a kind of a great book I'm trying to read right now about one of the researchers who did this research. She a lot of it comes down to trial and error. You don't know exactly where these enzymes are going to cut, so you gotta just start researching. And that's a good metaphor for life. Sometimes in life you just totally gotta go for it. All right, that's enough for part 1.